Good day, everyone. I would like to thank the PVMA for giving me the opportunity to present my research to you today. So, I'll be talking about some zoonotic hookworms that we should be thinking about in the Philippines. And similarly, I'll also be talking about benzimidazole resistance, which I fear and my research colleagues fear are becoming more and more a problem in the Asia Pacific region and also in the Philippines. So for this presentation, I declare no competing interest, and my interest in engaging in this research is primarily academic because I am a PhD student currently at Concan University. And this presentation is similar to like a literature review, and there will be researches that will be mentioned. And if you are interested in each of them, you may feel free to dig deeper because each of them will be cited in the PPT slide. So if you're interested, please do look into them individually. And because at some point in this presentation, I will be mentioning some formulation of benzimidazole drugs that are currently available there in our country. And I just want to declare that I am not linked to any, or I am not employed by any veterinary drug company. And yeah, those are my declarations and disclaimers. So in this presentation, we'll be looking into Ancylostoma ceylanicum and other zoonotic hookworms. I'll be looking into their epidemiology. Where are they occurring in the Asia Pacific region, or have they occurred in our country? Then we'll look into the molecular epidemiology. Primarily, I am interested in discussing with you the transmission dynamics of this zoonotic hookworms. Then lastly, we'll go into the benzimidazole resistance and why am I doing my research in this and why I think this is an emerging problem in our country. So we'll discuss on that and we'll end on some research gaps and future directions that we may engage in research. So among zoonotic hookworms, the literature says that they are still quite prevalent globally, especially in the Asia Pacific region, where they are said to cause around 500 million cases globally and 800,000 to 4 million disability adjusted life years lost. And it has been noted that a large sum of money is put into their control and elimination, including human elimination is human as a public health concern and eliminating the risk in veterinary medicine, especially in companion animals. So, among these zoonotic hookworms that we know, of course, number one of which is Ancylostoma ceylanicum. Oh, we also have Ancylostoma caninum, so I hope everyone is quite familiar with that one. So it's the it is zoonotic, but it's the common dog hookworm. Ancylostoma brasiliense, Ancylostoma tubiforme, and Poncinaria stenocephala. So as you can see in here, primarily in the Asia Pacific region, they have been noted, these hookworms have been noted to cause almost 100 million cases yearly. And in the case of Ancylostoma ceylanicum, this is a hookworm of dogs. And it has evolved through the close interaction of dogs and humans, it has evolved to become zoonotic. And oftentimes, because we commonly diagnose hookworms through fecalysis, so we only see the eggs. And if you can still remember our parasitology classes, we cannot speciate or we cannot identify the species only through egg morphology. So sometimes these are misdiagnosed. So we see a hookworm in dog, and sometimes we just think that, oh, it's Ancylostoma caninum. But as I will show later on, oftentimes these are the zoonotic Ancylostoma ceylanicum. And as you can see in the picture or in the figure on the right, I believe, yeah. So they are occur pretty much almost everywhere in the Asia Pacific region, including the Philippines, which I will be talking about later on. Now, for the life cycle, we know that the hookworms are their life cycles are direct. So there's no snail intermediate host. It's only the human or the animal, which is the final host where the maturity occurs. And you have the immature stages that are presently that can be present in the environment, such as the soil. So usually, um, the pre-patent period or the period from infection through egg passage to the feces usually would commence or would happen within a single month or a month and a half, but usually within a month in humans. But in dogs, it occurs quite faster. In 2 weeks, it can actually, upon infection, after 2 weeks, it can release thousands of eggs per day. So imagine if you have dogs around that are not dewormed regularly, they would release a lot of eggs, and a lot of these eggs would mature and become the infective stage. And if you have children that are not wearing their proper footwear, or you have children playing in the soil, they may become infected with these hookworms that come from animals. Now, looking at the transmission dynamics, so when we say one way of looking at transmission dynamics is through assessing the molecular epidemiology, or in this particular case, the genes that are um, usually we use the mitochondrial genes, for example, COX-1, and we look at the similarities of the genetic constitution or genetic composition of that particular gene from hookworms that are from different hosts. So, as you can see on the left side, yeah, this one, you have isolates from Thailand, so Southeast Asian nations, and when they do phylogenetic analysis and they construct a phylogenetic tree such as this one, you can see that there are two clades that are arising. So you have the zoonotic clade. So these are samples from both humans and animals. So they group together, meaning the hookworms from humans and animals are quite similar. And you also have human-specific clade or the human-specific group. On the other hand, we also have some research saying that they may also be a zoonotic haplotype and an animal-only haplotype. So what does this mean? So when we say that, for example, the mixed clade or the zoonotic haplotype, so if the genetic composition of the hookworm from the human or and from the animal is quite similar, so it's a pretty good hint that it may be a zoonotic infection. And if we have these three clades, when you put them together, and to simplify it, it will look like this. You have a bigger group or a bigger group of the hookworms of the Ancylostoma ceylanicum that occurs within this big zoonotic clade, which may be circulating between humans and animals. And similarly, you also have two other clades that are smaller in proportion, according to present research, that is only circulating between animals and also humans. Now, the question is which of these are present in the Philippines? And as of the moment, we do not know. We have evidence that Ancylostoma ceylanicum is present in the Philippines, but there has been no phylogenetic or any further molecular epidemiology, molecular biology, or genetics assessment of our Ancylostoma ceylanicum isolates. And hopefully, I will be the one to answer that question. But as of the moment, I cannot answer that question just yet. Now, looking into the pathological consequences of infection, of course, as I've said earlier, so you can acquire infection through contact with the environment wherein the infective stage is present. So, for example, soil. So, if you go out and if you do not wear your shoes, and if the environment is contaminated with the infective stage, you may get creeping eruptions or cutaneous larva migrans. So it's basically the infective stage burrowing into the skin to get into the blood circulation. Also, as you can see here, Ancylostoma caninum can cause eosinophilic enteritis. Eosinophilic enteritis in humans because yes, the dog hookworm can cause infections in humans, and because the immune response of the humans is quite strong to these dog hookworm infections, they cause a big inflammatory response causing eosinophilic enteritis, as you can see here. Then you have Loeffler syndrome. Loeffler syndrome, when the worms is traveling through the lungs, the upper respiratory system, for them to be swallowed. So sometimes it causes respiratory problems. So now, inuubo yung worm papunta sa GI tract. So that's the Loeffler syndrome. And of course, most commonly, we do associate hookworm infections with anemia, specifically iron deficiency anemia. And as you can see here, our concern as veterinarians is of course in dogs. And as you can see, almost half a liter in heavy infections, almost half a liter of blood can be lost in 14 days. So imagine a heavy infection in a puppy, which will lose approximately 500 ml per 14 days. So that's going to be a big problem, specifically when you check the blood parameters through CBC, for example. Now, an emerging issue, or not really an emerging issue, but this becomes a public health problem when a large number of susceptible individuals, so for example, children, school-aged children, are infected with a lot of worms in a lot of areas. And this happens in the Philippines, where STH, soil-transmitted helminths, including hookworms, are quite prevalent. So they cause a lot of issues in children which may have long-lasting effect. For example, impaired cognitive and motor development. So they have long-lasting effects even though the infection occurs in younger children. Now, looking at Ancylostoma ceylanicum, at first, as you can see, as the years say, Ancylostoma ceylanicum in the early 20th century, I believe, is not actually a really big problem. So, because the main thing was we did not know that it is a big problem because we are confusing it, misdiagnosed, misdiagnosing it with other hookworm species because what we use for diagnostics is mainly through egg detection and egg identification. Now, in the early 2000s, through the rise of molecular techniques such as PCR, and as this technique became more specific to identifying species, we started to learn some interesting things about Ancylostoma ceylanicum. And up to the present, it's still considered an emerging disease because we have good evidence so far, but we need more evidence, specifically in their epidemiology and geographic occurrence. Now, another issue that I think veterinarians concerned with public health is we need to be considering these results, recent results at that Ancylostoma ceylanicum being present in a country which is reliant on tourism, it becomes a problem because it may cause travelers diarrhea, as you can see in the several research. And we know that our country is very big, very much big on tourism. So we may have an issue in our hands considering that Ancylostoma ceylanicum is zoonotic. Now, why am I making a big deal with Ancylostoma ceylanicum? I'm making a big deal out of this zoonotic hookworm because there are a lot of evidence right now pointing out that Ancylostoma ceylanicum is actually number two hookworm, the number two hookworm in the Asia Pacific regions among humans. So when we say human hookworms, what we think of normally in tropical medicine is Necator americanus and Ancylostoma duodenale. But through the advancement of diagnostic methodologies and being able to identify the species of these hookworms from field isolates, we determined, or the researchers have determined, that Ancylostoma seylanicum is number 2. But that's in 2012. How about more research, more recent papers? Still the same. As you can see in this recently published paper from Vietnam. Number 2 is Ancylostoma seylanicum, 31%. Second only to Nicanor americanus, which is 47% in that particular area. Now, the question is this occurring in the Philippines? So before I answer that question, let's look into hookworms. Let's look into hookworms in the Philippines. So our recent research on national prevalence data on soil-transmitted helminths. So they study ascaris, trichuris, and hookworms. So our recent reports say that the prevalence of hookworms in the Philippines among children, school-aged children, is actually less than 1%. So it's significant but not quite. And as you can see, in the geographic distribution of the hookworm infections, it's mainly occurring in Visayas, and mostly the more prevalent areas are in Mindanao. And among the in the among to the veterinarians from Mindanao, I think you can see why. But the problem is, this may be underestimated because they are using fecalysis technique, specifically catocats. So looking at this evidence, one may say that it's less than 1%, so why does it matter? So does it really matter? But I would argue otherwise. Now, recent research in the Philippines conducted by, for example, the group of Professor Catherine Gordon from Queensland, University of Queensland. So they were using PCR techniques, and as you can see, it's not less than 1%. Although these research are conducted in specific areas, for example, in Leyte and Samar, but nonetheless, they are saying that it's nearly 50%. And if you look here, yes, Ancylostoma seylanicum seems to be the number 2 hookworm in the Philippines, next to Necator americanus, similar to the research from other countries that I presented previously. So the problem is, what's the epidemiology of Ancylostoma seylanicum in the Philippines? Where does it occur? What are the risk factors for it? And does the animals in the same areas carry the Ancylostoma seylanicum also, implicating zoonotic resistance? That we do not know. And that research question is part of my PhD research currently. So hopefully I'll be able to deliver the results to you on a future date. But what I can share to you right now is that we have run some samples from Samar and Bohol, and indeed, I can share with you that through assessing the PCR assessment of COX-1 gene of Ancylostoma seylanicum, we have human samples being positive currently in Samar and from Bohol. These samples are from Dr. Angeles from the UP College of Public Health. So indeed, Ancylostoma seylanicum is still present in the country. However, these samples are currently being sequenced, and so I cannot tell much on their genetic composition right now. Now, looking at animal infections. So let's go back to this paper from Cambodia. And as you can see, Ancylostoma seylanicum is actually number 1. So it's not Ancylostoma caninum. And also here, this results from Bangkok, from the group of Professor Tawin Ipengkao from Kasetsart. And as you can see here, 70% of their positive samples were seylanicum, saying that Ancylostoma seylanicum is the number 1 hookworm in the Asia Pacific region. So that's our hypothesis right now. Now it's very easy. So I hope you can put one and two together. It's number two in humans and number one in dogs and or in animals in general and companion animals to be specific. So if you put that together, it's not impossible to think that it's number two in humans because the source is animals, as I've said. Now, our issue with this one is that the things that I have told you or the things that I have said is yes, it is based on research, but we do need further evidence for this, specifically in our country. So, yeah, I think it's this venue, the PVMA, is a good platform to advocate that we need attention or we need to put attention in zoonotic hookworms, specifically zoonotic hookworms coming from companion animals, it doesn't, which poses a risk to public health, especially to dog owners. Now, let's zero in. Speaking of hookworms in our country, what do we know so far? So, we have a lot of historical evidence of zoonotic hookworms. So, yes, Caninum, we know that already. The Stomacaninum, of course. And surprisingly, we have previous reports of Ancylostoma brasiliense, which is my students would say that it's not present because Braziliensis it's from Brazil. No, we have had them in our country, and they are the main cause of creeping eruptions. So we have a lot of hookworms in our country, to say the least. And to end this part, speaking about hookworms, so I have published this commentary. It's actually an opinion paper or an editorial paper which is the main point I'm saying is that it's very common. I think a lot of veterinarians, both in public health research or in parasitology or in companion animal medicine or companion animal practice, would say that yeah, it's very common with prevalence. Even reports in peer-reviewed papers, 8 to 48%. So it's not far-fetched to say that it's very common. I believe. And the problem I had, why I wrote this paper, why I said it's very much neglected in veterinary research, is that, as you can see, from I believe 2010 onwards, we only have 5 papers. And um, I feel I seem like I'm venting out, but medyo nakakahiya as an international student from in another country doing a PhD. Questionin ka ng mga professors mo, you only have 5 papers from your country. So, I do believe that I think we need to step up in researching zoonotic hookworms. Even just the basic epidemiology research, I think we need to publish it in peer-reviewed literature so at least we who are researchers can get a grasp of the occurrence in the epidemiology so we can help our fellow veterinarians, so our companion animal practitioners to help them in developing control and elimination programs for this particular concern. And we can also input in policy within the human aspect because, for example, as Sumaselanicum, as I have said, it occurs in the Philippines, but the problem is we don't know where. Aside from this research in Samar, aside from that, we do not know where they occur. So that's a problem because again, it is zoonotic. Now, moving into benzimidazole resistance. So, as benzimidazole, as the name imply, yung may mga apelyedong dazole sa pangalan. So, albendazole, fenbendazole, and others. So, what they usually do is just a review of pharmacology. So, they inhibit tubulin dimerization. So, si tubulin is composed of alpha and beta tubulin. So, mag-bind silang dalawa to form the tubulin dimer. Then the tubulin dimer will be the ones to polymerize to form the microtubule. So I hope you can still remember your cellular physiology. to know that microtubules are part of the cytoskeletal structure. So they perform a lot of important functions in the cell, not only mammalian cells but also in parasitic worms. So when you inhibit this one, you essentially kill the worm. So albendazole does that. It inhibits the polymerization of the microtubule. Now, benzimidazole resistance in hookworms are caused by mutations in the beta tubulin gene. So these mutations primarily occur in specific locations. So for example, amino acid 167, 198, and 200. And among canine hookworms, specifically Ancylostoma caninum, these mutations, resistant associated mutations have been reported in several areas in the world. For example, in Brazil, a lot of it is in the United States, as you can see. And the prevalence of these mutations could actually be as high as 49%. So it's quite prevalent. And most recently, we have a we have a paper from Australia. So it's in the Asia Pacific region already. Several of these single nucleotide polymorphisms. So these are base pair changes that alter. Basically, the point is you make the protein sequence different so that the drug cannot bind to it. And you can so that there's that's the resistance mechanism. The drug will not bind to the protein, so the protein can do its function normally. So polymerization is not inhibited. Now, in our recent literature review, we found that among STH, or lahat na ito, soil-transmitted helminths, ascaris, recurids, and hookworms, we found that a lot of research has been published on these single nucleotide polymorphisms. Specifically, they were conducted in the Americas, in the American region, and in African region. So STH in humans. And as you can see, there's a big blank here in the Asia Pacific region, except the ones that I mentioned previously. So also one question that I am asking in my PhD research is that does these SNPs or does these mutations, resistance associated mutations occur in the Asia Pacific region, specifically in hookworms in Southeast Asia? And one way of not answering, but one way of looking into the effect of these SNPs is through in silico studies or computational biology. And here, I would like to share one of my first results in my study. I hope you can see, but I think you can see. But the thing is, through our literature reviews, so I had two literature reviews that are currently under review. They're submitted in a journal. We found that there are 7 mutations in these 4 locations. So 134, 167, 198, and 200. These positions are amino acid positions. So these are the changes. So for example, in amino acid 134, it's originally glutamine. But through the mutation, nagiging histidine siya. So the point is to change the structure of the protein so the drug cannot bind. So in this study, we did in silico studies, so a lot of computational biology. So what we did was we input these mutations, we dock. So in silico docking, we dock the protein and the drug. So in this particular case, I think this is fenbendazole. So we drop it inside the binding site, look at the interactions of the amino acids. So anong mga amino acids ang nag-i-interact in silico, and we look at binding affinity. So as you can see, I hope you can see here, there are certain mutations, particularly 198 and 200, when glutamic acid becomes lysine and when phenylalanine becomes leucine, the binding affinity or the binding efficacy drops. So meaning the drug is not binding properly to the binding site. So which potentially causes resistance. So here we provide evidence in silico that these mutations do alter the binding affinity and the binding interaction of the benzimidazole drug and the beta tubulin. So hopefully this will get published soon, very soon. But anyways, here, I think this is the buod, kumbaga, the buod of my talk. And why is this important? Because when we talk about zoonotic hookworms, of course, we're talking about transmissions between humans and animals. And one thing we need to know is that sa humans, I know a lot of you public health veterinarians know that our DOH have a control program for STH. It's the Integrated Helminth Control Program that started in 2006 and it's still currently ongoing as to this day. So what do they do? So to control STH infections, they administer benzimidazole drugs, to be specific, albendazole or mebendazole. So depende na lang sa prevalence kung ano how often are they administering this through school-based mass drug administration. Now, partner that in veterinary medicine, of course, we're very good at deworming our patients. And I hope a lot of you know this by now. Natatakpan ko siya. But the point is, there are a lot of recommendations right now from trusted sources such as the Tlocap saying that we need to deworm our dogs or our animals on a monthly basis. So when I was in vet school around 3 years ago, no, not 3 years, yeah, 29, 4 years ago, what we were taught was we need to deworm the dogs for regular deworming. We need to do it on a quarterly basis, so every 3 months. But currently, the recommendation is to do it monthly. And a lot of our drugs also in the Philippines also contain benzimidazole derivatives. So you have a situation here wherein on the human and the animal side, the hookworms can be exposed to benzimidazole drugs. And yung exposure na yun can put positive selective pressure for the worms to become mutated or to acquire mutation that will help them survive the drug administration. So those mutations that I've said before, mutations that occur in the beta tubulin gene, yung mga pinakita ko before. And as you can see, checking out the list of our drugs currently, I got this list from PVET, I believe. Yeah, PVET. And as you can see, those in red arrows are the ones containing benzimidazole derivatives, primarily fenbendazole and febantel. And what's surprising or what's alarming to us is that you can actually buy it in Shopee without a veterinary prescription, which remains a big problem in our country, I believe. And yeah, this adds to my fear that we are fast tracking ourselves to multi-drug resistant or even benzimidazole resistant hookworms, which has been a big problem in the US right now because a lot of our drugs contain benzimidazole drugs and even humans, as I have shown in the previous slide, even humans use benzimidazole drugs. So we are in a situation wherein even if the worm gets the mutation or acquires mutation in the animals through repeated exposure to the drugs, if it is a zoonotic hookworm, it may find itself infecting a human. And at the same time, through mass drug administration to control STH in humans, it may get the resistance there and also find its way to infect and to become resistant in animals so that veterinary interventions will not be effective anymore. So that's a concern that I am quite worried right now. So what about research gaps? So to end, this is I think my concluding slides, but to end, we do not know zoonotic hookworms much in the Philippines. We're quite familiar with them. We've met them, but we don't really know where they are. And that's a problem because when we have something that we do not know, it may return to haunt us at the end. And yeah, studies of molecular identification and molecular epidemiology really need attention, not only for zoonotic hookworms, mind you. A lot of other parasites and a lot of other microbes or pathogens need to be assessed I think one of the good part of ASF becoming a big problem in the country is that a lot of researchers have come into the lab, have got their bootstraps together and looked into the molecular genomic, the genomics level of hookworms, genomics level of the ASF and AI, so and rabies actually. So hopefully we would be able to do that in our parasites that are endemic in our country. So yeah, molecular epidemiology transmission dynamics. This is quite important because we need to control or at least eliminate and prevent zoonotic hookworm infections through policies. So we need evidence. We need further studies on that one. And of course, benzimidazole resistance, as I have said, um, we are on the same side, same two sides of the same coin. So whether resistance occurs first in the human side, it may have a problem. It may cause havoc in the animal side, or even if resistance occurs in the animal, in the dog side, or in the veterinary side, if it's Ancylostoma seilanicum, then we may have a problem with the human side because the control program will not be effective anymore because the control program is reliant on mass drug administration. So I hope in this talk I was able to bring attention to zoonotic hookworms and benzimidazole resistance, which may potentially become a problem in our country. So I believe being proactive is better than being reactive. So I think it's good that PVMA gave me the opportunity to highlight these particular emerging problems in our country. So thank you very much once again. I am Dr. John Kaiden B. Tenorio. So currently I am a PhD student in tropical medicine at Konkan University here in Thailand. And yeah, I would like to thank, of course, the Tropical Disease Research Center, which I belong currently, and my home university, the CVM at University of Southern Mindanao. And thank you so much to the Philippine Veterinary Medical Association for giving me this platform to bring awareness to the causes or to the issues that I highlighted in this talk. So if you have any questions, um, please do not hesitate to email me or contact me through these social media platforms. I hope makita ninyo. But anyways, you can send me an email at these email addresses. And I'm very sorry that I cannot be there in person. I was planning to be there, but there were some issues with my research that I currently need to focus on. But anyways, if you do have any questions, just hit me an email and I will get back to you. So thank you very much. Hapunahap.